So I want you to just imagine for a second that you are standing in the middle of a massive, just sprawling library.
Oh, okay.
Yeah, and there are, millions of books and billions of pages stacked from the floor all the way to the ceiling.
Right.
Now, imagine your job is to find a single specific typo, just one misspelled word hidden somewhere in that entire ocean of paper.
That sounds absolutely impossible.
Right. But that is essentially the challenge of modern cancer detection when we use liquid biopsies. We are searching for incredibly rare tumor mutations, that one specific genetic typo floating in a vast sea of healthy DNA in your bloodstream.
It is an incredibly daunting task, and honestly, it's one of the biggest bottlenecks in modern oncology right now.
Really? Why is that?
Well, we generally like to think of medical diagnostics as binary, right? Like a broken bone on an X-ray is either there or it isn't.
Yeah, makes sense.
But looking for these microscopic genetic typos requires something called ultra-deep sequencing. You have to read the exact same stretches of DNA tens of thousands of times just to be absolutely sure the typo is actually there and not just a machine error.
Oh, wow. Tens of thousands of times.
Exactly. It's highly precise, but it comes with massive drawbacks. It's prohibitively expensive, for one, and it completely misses cancers that simply don't shed many mutations into the blood. Plus, because of the cost and complexity, it severely limits how widely we can actually screen the general population.
Which is exactly why the 2026 study we're exploring today in this deep dive is so fascinating. It's from the journal Cell Reports Medicine.
Yeah, the Fragmentia-AI paper.
Right. The researchers developed an AI model called Fragmentia-AI. And what really caught my eye here isn't just that it's a new algorithm. I mean, we see those all the time. But it represents a total paradigm shift in how we actually view biology.
It really does. It's a completely different way of thinking.
Because instead of treating DNA like a rigid genetic blueprint where we are just obsessively hunting for that specific typo, they're treating DNA like a language.
Yes, exactly.
And the mission of our deep dive today is to explore how looking at the grammar of our biology could completely democratize how we detect cancer.
Yeah.
Like how we monitor a patient's recovery and even how we predict relapses.
It's huge. But to really grasp how an AI reads DNA like a language, we have to start with what is actually floating around in your blood when a phlebotomist draws a vial, because it's not just whole, perfectly intact cells in there.
Right. You have something called cell-free DNA or cfDNA. And the way I visualize this is almost like exhaust fumes coming off a running engine.
So I like that analogy.
As the cells in your body go about their business, they eventually age and die. And when they die, they break apart and shed tiny fragments of their DNA directly into your bloodstream. It's a completely natural process, right?
That's a great way to put it, yeah. The body is constantly turning over cells, so there's always a baseline level of this cfDNA in your blood. Okay.
Now, if someone has a tumor, those cancer cells are also dying and shedding fragments. That specific tiny subset of fragments is called circulating tumor DNA, or ctDNA.
And if I'm understanding the research correctly, this is exactly where the old typo hunting method just runs into a brick wall. The study points out a pretty staggering fact, actually.
What's that?
That only a tiny minority of those circulating tumor DNA molecules actually could carry the known mutations we are looking for.
Yes. That is the crucial limitation. Current tests throw away the vast majority of the sequencing information they gather. They are entirely obsessed with finding that one specific genetic mutation.
Wow.
Yeah, so if the DNA fragment they pull from your blood doesn't have the mutation on it, the test essentially ignores it.
So it's like finding a torn, crumpled page from a book, but because the specific typo you were warned about isn't printed on that exact page, you just throw the whole thing in the trash.
Exactly.
You're completely ignoring the fact that the page is violently ripped and clearly came from a damaged book. Yes. And that realization is what birthed the whole field of fragmentomics. Instead of just looking at the sequence of the genetic letters to find a mutation, scientists started looking at the physical structure of the fragments themselves.
Like the actual shape and size of the trash.
Right. They look at the link of the fragment, where it was cut, and the overall chaotic patterns of how it was packaged. Because tumor DNA is packaged and degrades very differently than healthy DNA. It's totally disorganized.
Okay, that makes sense.
Previous models, like DEAL-FFI or Griffin, they tried to capitalize on this by using what we call engineered features.
Wait, let me stop you there, because that term engineered features feels important to unpack for everyone. That basically means human-defined rules. right?
Yes, exactly.
Like a human programmer sits down and explicitly tells the computer, hey, I want you to count all the short fragments, or I want you to specifically look at the ends of the DNA.
Precisely. The humans were still acting as the translators, setting the parameters based on what we thought was most important.
But the paper says Fragmentia-AI throws that entire rulebook out the window. So if we aren't giving the AI any rules, how does it know what to look for? I mean, how does treating DNA like a language actually work in practice?
It works because Fragmentia-AI uses large language models, or LLMs. This is the exact same underlying architecture that powers the advanced text AI we use every day on our phones.
Oh, wow. Okay.
These models don't need human-defined grammar rules. Think about how a toddler learns to speak. You don't sit a 2-year-old down and teach them the rules of past participle verbs, right?
No, definitely not.
They just listen to thousands of hours of conversation, and their brain naturally picks up the underlying syntax. By processing millions of raw DNA fragments, Fragmentia-AI learns the intrinsic language or grammar of tumor DNA directly from the raw sequence. Nobody tells it to look for short fragments. It just learns what the language of cancer inherently looks like.
So the AI is essentially learning the dialect of a tumor just by exposure. That's wild. Let's do a quick recap here for you guys listening, just to lock this in before we go deep.
Good idea.
Okay, so bullet 1, your blood contains fragments of DNA called cfDNA. And a tiny bit of that is from tumors, which is ctDNA.
Right.
Bullet 2, old methods look for rare mutations, which is super expensive and misses a lot of cancer. And bullet 3, Fragmentia-AI looks at the structural grammar of the DNA fragments using language models instead of human rules.
Perfect summary.
Awesome. So moving on, if that's the case, I really want to get into the mechanics of how a computer actually reads a biological molecule. Because a DNA sequence isn't written in English, it's just billions of A's, C's, T's, and G's.
Yeah, so the first step is translating that chemical sequence into something the language model can actually digest.
Yeah.
They use a process called DNA byte pair tokenization.
Byte pair tokenization. Okay, unpack that.
Well, in traditional genomic analysis, you read the DNA letter by letter, but byte pair tokenization groups those letters into meaningful biological syllables or words, which we call motifs.
Oh, okay. So it's the difference between sounding out a word letter by letter like CAT versus just seeing the word CT and instantly conceptualizing a feline. The AI is chunking the DNA sequence into biological words.
Yes. And that chunking is brilliant because those biological words are then fed into a transformer model, specifically one called DNABERT2. The core mechanism that makes a transformer so powerful is something called self-attention.
Self-attention. I've heard that term in AI a lot.
Yeah, in human language, context is everything. The word bark means something entirely different if the surrounding words are about a dog versus a tree.
Right. Totally changes the meaning. So self-attention does the same thing for DNA.
Exactly. The biological meaning of a specific genetic motif changes depending on its neighbors. For example, is this specific motif sitting right at the edge of a fragment where a biological enzyme cleaved it from the main genome? The AI's self-attention mechanism looks at that context to figure out the structural reality of the molecule.
Hang on, I'm stuck on something here. Let me play devil's advocate.
Go for it.
If we go back to the blood draw, a patient's blood sample has millions of these DNA fragments. And if I'm an early-stage cancer patient, the vast majority of those fragments, like 99.9% of them, are totally healthy exhausts from normal cells.
That's correct.
So if you feed millions of healthy fragments and just a tiny handful of tumor fragments into an AI, wouldn't the cancer signal just completely drown in the noise?
It absolutely would if the model simply averaged the signals together. Averaging 1 million healthy signals with 10 cancer signals would dilute the cancer signal down to essentially 0.
Right. So how do they fix that?
To solve this, the researchers utilized a mechanism called multiple instance learning, or MIL.
Okay, let me try an analogy here to see if I've got this. Tell me if this tracks. Let's say a detective is handed a massive garbage bag full of evidence from a crime scene. Most of the items in the bag, a grocery receipt, a discarded piece of gum, a crumpled napkin, they're totally irrelevant to the crime.
Okay.
But buried at the very bottom is a single shoe with a bloody footprint. The detective doesn't look at the bag and say, well, on average, this bag is mostly harmless trash. They ignore the receipts and assign massive importance to the footprint.
That is exactly how multiple instance learning functions. The AI treats all the fragments from a single patient's blood draw as one bag of evidence. The MIL mechanism evaluates every fragment, but it assigns much higher mathematical weight, what we call attention weights, to the most suspicious fragments.
So it's picking out the bloody shoes.
Right. It effectively plays detective, zooming in on the tumor signal and isolating it from the healthy noise. From there, it generates a single prediction score for the whole patient.
Okay, conceptually that makes total sense. But logistically, evaluating millions of fragments per patient across thousands of patients, that sounds like a computational nightmare. If I tried to run that on my laptop, it would melt.
It would absolutely would.
And even with big servers, wouldn't the AI just end up memorizing the data rather than actually learning the biology?
Yes, memorization is a massive risk in machine learning. We call it overfitting. The AI just memorizes the answers to the test rather than learning the concepts. To prevent that, the researchers introduced something called stochastic pool sampling, or SPS.
Stochastic pool sampling, what does that do?
During the training phase, out of the up to 12 million fragments available for a single patient, they randomly sample just 3,000 fragments per cycle, or epoch.
So they are constantly shuffling the deck.
Yes, they show the AI a new random subset of 3,000 fragments every single time it reviews a patient's profile.
This is wild because the source material actually includes an ablation study on this exact mechanism. And for you guys listening, an ablation study is basically where scientists turn off a specific feature of the AI to see if the whole thing breaks.
A stress test for the algorithm itself.
Right. And when they turned off that random shuffling and just fed the model a static set of fragments, the model completely collapsed. It memorized the training data. And when they tested it on new patients, its accuracy plummeted to a coin flip.
It became totally useless.
But with the shuffling turned on, it never overfit. By constantly forcing the AI to look at a new random sample, it forces the system to learn the actual underlying biological rules of cancer.
It's a beautifully elegant solution to a massive computational problem.
All right, time for another quick recap before we get to the real-world results. So, bullet 1, DNA is chunked into words using byte pair tokenization.
Yep.
Bullet 2, Transformer AI reads the context of these words using self-attention. Bullet 3, multiple instance learning flags the most suspicious fragments in the blood. And bullet 4, stochastic pool sampling keeps the AI from memorizing the data by mixing up the training fragments.
Spot on.
Okay, so at this point, the AI engine is built. It knows how to read the biological words, it knows how to play detective, and it's trained not to cheat. The researchers trained Fragmentia-AI on nearly 3.7 billion unique sequences from lung cancer patients with high tumor signals. But training an AI in a lab is one thing, right? The real test is how it performs out in the wild against the gold standard.
Exactly. The clinical stress tests are where this study goes from just an interesting computer science paper to a potential medical breakthrough. The first major hurdle was seeing if the AI could detect cancer when the tumor signal in the blood was incredibly low. We're talking less than 0.5% tumor DNA in the sample.
And it gets even more extreme than that. They ran this AI on a specific group of 75 lung cancer patients who had 0 detectable tumor mutations.
Yes, 0.
If you use the absolute best, most expensive, ultra-deep sequencing available today to look for genetic typos in these patients' blood, you would find nothing. The gold standard tests were completely blind to the cancer.
By traditional metrics, those tests would tell the doctors that the blood was totally clear. But Fragmentia-AI detected the cancer in those mutation-negative patients with an AUC of 0.891.
Which is crazy. And just for context for the listener, AUC is an accuracy metric where 1.0 is absolute perfection and 0.5 is a random guess.
Right.
Hitting nearly 0.9 on patients where the traditional test saw absolutely 0 evidence of disease is incredible. It found the cancer purely by reading the structural grammar of the fragments, completely bypassing the need for a genetic typo.
Which naturally leads to the second and arguably more globally impactful stress test, the pan-cancer cohort. This gets to the heart of why current tests are so ridiculously expensive.
How so?
Well, standard mutation tests require a sequencing depth of about 30,000x. That means the machine has to read the exact same stretches of DNA 30,000 times over to confidently identify a rare mutation.
Wow, 30,000 times.
Yeah. But this study tested Fragmentia-AI on data sequenced at an ultra-low depth of just 0.1x.
Wait, 0.1x. That means they're using a fraction of a percent of the conventional data input. The AI is practically starved of data compared to normal tests.
It is extremely sparse data. And they didn't just test it on lung cancer this time. They unleashed the model across 17 different cancer types: breast, liver, sarcoma, melanoma, ovarian, spanning over 2,000 patients.
And how did it do?
It reliably distinguished the cancer patients from the healthy controls with a really impressive global accuracy. The AUC was 0.885.
Okay, I have to jump in here because I'm genuinely skeptical about something.
Okay, what is it?
If you take a machine learning model, you drastically change the sequencing equipment, you starve it of data by dropping the input to 0.1%, and then you throw 17 new types of cancer at it. How do we know the AI isn't just hallucinating?
Hallucinating.
Yeah, like how do we know it didn't just find a weird technical artifact or a glitch in the ultra-low depth sequencing machine itself, and it's just exploiting that glitch instead of actually finding biology?
That is the exact right question to ask, and honestly, it's the nightmare scenario for any data scientist. To prove it wasn't just a machine hallucination, the researchers ran a reciprocal validation experiment.
Reciprocal validation, what does that entail?
They took a version of Fragmentia-AI that was trained exclusively on that sparse ultra-low depth data. They essentially raised it in that specific technological environment. Then they took that model and tested it on the deep, targeted panel data from the original lung cancer cohort. These are two fundamentally different chemical assays run on different machines with completely different background noise profiles.
Oh, I see.
If the AI was just memorizing a machine glitch, it would have failed instantly on the new machine.
But it didn't fail.
No, it didn't. It maintained a high level of accuracy. That reciprocal validation proves the AI is detecting universal biological truths about the structure of cancer DNA. The physical chaos of tumor DNA is a shared language across all those different malignancies, regardless of what machine you use to read it.
That's amazing.
Furthermore, it statistically outperformed 6 other leading fragmentomic models on the exact same data.
Because those older models were still using the human-engineered rules, right? They were still throwing away the contextual information of the individual molecules, whereas Fragmentia-AI is reading the entire biological sentence.
Exactly. But detecting cancer in a controlled study is just step one. If you're a patient listening to this, the most pressing question isn't just, can an AI find cancer in a database?
Right, it's how does this actually change my treatment?
Exactly.
And let's do our third quick recap here. So the AI detects cancer even when traditional mutations are completely undetectable. It works across 17 different cancer types, proving a universal cancer grammar. And it only needs a fraction of the sequencing depth, slashing costs.
Perfect. So going back to treatment.
Right. If you or a loved one just finished a grueling surgery to remove a tumor, or you just wrapped up months of immunotherapy, the absolute biggest question hanging over your head is, did we get it all?
That anxiety is tied to what oncologists call minimal residual disease, or MRD. Even after a successful surgery, microscopic clusters of cancer cells can remain hidden in the body, slowly growing until the cancer relapses.
It's a terrifying thought.
It is. The researchers used Fragmentia-AI to analyze the blood of lung cancer patients right after they finished their treatments.
And the predictive power was stark, wasn't it? The data show that patients who were flagged as MRD negative by the AI, meaning the model, could no longer hear that cancer language in their blood had significantly better survival rates without recurrence.
Yes. It could reliably predict who was likely to relapse and who wasn't, weeks or even months before a new tumor would ever be visible on a traditional CT scan. It offers a crucial window for doctors to intervene earlier.
That is life-altering information.
Yeah.
But it brings up a philosophical issue I have with AI and medicine. We know the model works, we know it accurately predicts relapse. But can the doctors actually see what the AI is looking at? Or is this just a magic black box where a blood sample goes in, the computer whirs, and a life or death diagnosis comes out?
That's a huge concern.
Because I can't imagine a doctor making a massive treatment decision just because a computer said, trust me.
The interpretability of AI is a massive focus right now, and thankfully, the researchers were able to pry open the black box. They extracted the specific mathematical weights the AI was using and looked at the physical properties of the top 1% of fragments, the ones the AI flagged as the most highly suspicious.
The muddy footprints in the evidence bag.
Precisely. And what they found perfectly aligns with what we know about the biology of cancer. First, the fragments the AI focused on were measurably shorter. The top cancer fragments averaged 154 base pairs in length, while healthy DNA fragments averaged about 166 base pairs.
Okay, so they're shorter. But the most fascinating part of what they found inside the black box has to do with how the DNA is actually cut. The researchers call it the attention smear. Let's talk about that.
Yeah, the AI was paying massive mathematical attention to the absolute ends of the fragments. This makes perfect biological sense because the ends are where cellular enzymes literally cleave the DNA apart when the cell dies. In healthy, normal DNA, the AI's attention at the end of the fragment is a neat, narrow, highly predictable spike.
I love the visual they use to explain this. Think of healthy DNA as being cut with a biological scalpel.
Right.
The process of healthy cell death is orderly. The cuts are precise, clean, and uniform. But when the AI looked at tumor DNA, that attention spike wasn't neat at all. It was a broad attention smear across the end of the molecule.
It's completely chaotic. If healthy DNA is cut with a scalpel, the AI is seeing tumor DNA that looks like it was chopped up with a rusty axe.
A rusty axe. That paints a picture.
Yeah, the ends of the tumor fragments are messy, disorganized, and highly variable. The AI specifically hunts for those irregular, jagged sequences at the exact spots where the DNA was severed.
It's essentially sensing a genome-wide shift in disorder. It isn't just looking for one loud signal or one specific typo. It's feeling the overall texture of the chaos. It mathematically recognizes the rusty axe.
And that transparency is what will eventually allow clinicians to trust the tool. It's not a black box. It's an incredibly sensitive pattern recognition engine looking at validated biology.
Let's do our final quick recap. Fragmentia-AI can predict if a patient will relapse after surgery or immunotherapy. And the AI proves it's working by focusing on biologically validated traits. Shorter fragments and messy, chaotic cleavage ends.
Exactly.
If we step back and look at the big picture, the implications of this shift from typo hunting to language reading are massive. But from my perspective, the most profound impact of Fragmentia-AI might not even be strictly biological. It might be economic.
Oh, absolutely. The economics of healthcare dictate what treatments actually reach patients. Because this model requires only 0.1% to 1% of the sequencing data compared to traditional mutation hunting, it fundamentally changes the financial math of cancer care.
Currently, population-wide early cancer screening using liquid biopsies is just too expensive to roll out for everyone. It's a luxury diagnostic. But by slashing the data requirement and bypassing the need for ultra-deep sequencing, Fragmentia-AI had the potential to democratize advanced diagnostics.
It really does. It makes population-scale early screening financially viable. It means a patient could get affordable, frequent blood tests to monitor for relapse during their standard clinical checkups, rather than waiting for an expensive yearly scam.
It shifts the liquid biopsy from a specialized, rare tool reserved for major cancer centers to something that could eventually be as routine as getting your cholesterol checked at your local clinic.
That's the dream.
But I want to leave you, the listener, with a final thought to mull over, something that builds on the limitations section of this very study. Right now, as incredible as it is, Fragmentia-AI is essentially a binary test. It tells the doctor, yes, there is a cancer language present, or no, there isn't.
But if you recall the multiple instance learning mechanism we discussed, the detective sorting through the evidence bag, the AI is incredibly good at mathematically separating the tumor fragments from the healthy ones. It assigns heavy weights to the chaotic fragments and ignores the healthy exhaust.
It's essentially digitally purifying the blood sample, isolating just the cancer data.
Exactly. So the next logical question is, what if we take that digitally purified pool of tumor DNA and use it to map the exact tissue of origin?
Wow.
Imagine going in for that cheap routine blood test we just talked about. The AI analyzes the fragments, but it doesn't just whisper, you have cancer. It takes those chaotic, rusty axe fragments, reads their specific dialect, and points exactly to your left lung or your liver or your ovary. It could tell doctors exactly where the malignancy is hiding in your body before a single symptom ever appears.
That is the true promise of treating biology as a language. We are no longer just searching for a misspelled word. We are finally learning how to read the entire story. Thank you for joining us on this deep dive into the hidden grammar of DNA. Keep questioning the world of information around you, and remember that sometimes the biggest breakthroughs come when we completely throw away the rulebook.